Okay, good morning. So thanks to the organizers for a wonderful conference and the invitation to speak, and particularly for the most awesome dance party last night. I'm sure you're all awake this morning. So I'm going to talk to you about cortical representations of pitch, which I've been working on for quite some time now. But I'm going to present to you this morning sort of brand new unpublished data that we have in my group, and focusing particularly on pitch for sort of more real-world listening. So Professor McPherson already explained to us on Tuesday that pitch is this tonal quality of the sounds that we hear. You can relate it to a musical scale if you're a human listener. And there are at least two sort of acoustical signatures that correlate with our perceived pitch. In the spectral domain, we have harmonicity. So the frequency components of the sounds that we hear are all multiples of the fundamental frequencies. So it seems that the auditory system binds all these frequency components, if they're harmonically related, to lead to one percept of a pitch. And this is the case across different instruments, different timbres, so there's no one frequency in here, that's the pitch we hear. And in fact, we can remove all energy at the fundamental frequency and we'll still perceive the pitch at that frequency. Now in the temporal domain, sounds that evoke pitches, even if they're produced across different instruments, are normally periodic. So these have waveforms that repeat at a rate of the fundamental frequency. So there are at least two acoustical cues that lead to this one unitary tonal percept that we experience. And I want to understand how the brain is able to integrate these cues and extract this percept that we have. So if we look into the auditory system, quite popular is a theory of dual pitch extraction. So psychophysical studies show us that you can either use these harmonic spectral cues or you can use the temporal cues. And there's a lot of evidence to suggest that these are actually processed in parallel in the auditory system to give rise to pitch. So in terms of the harmonic cues, you have auditory nerve fibers that are tuned. So you have an individual neuron tuned to an individual harmonic. So you end up with a place code for these harmonic relations. Up on the sort of higher end of the harmonics in the sound, you now have a neuron responding to a combination of harmonics. And so as they try to phase lock to the temporal envelope of that sound, they'll actually phase lock to F0. So now you have a spike rate code in these higher unresolved harmonics of pitch. And I sort of imagine that somewhere in the brain, probably the auditory cortex, you might have harmonicity neurons that are sort of set up with circuits to extract these resolved harmonic cues, and temporal neurons that are instead looking at the firing of spikes in time to extract the temporal cues. So that's going to be my sort of working hypothesis today. And you might even imagine that somewhere in the auditory cortex, perhaps higher in the auditory cortex, you would have a pitch neuron that can integrate these cues and use either or both to provide you with a perceptive pitch no matter what complex sound you're listening to. So what evidence do we have in animals for actually any existence of pitch representations or these harmonic and temporal representations? Well, neurons throughout the auditory cortex represent the fundamental frequency of complex sounds. And I would refer to these as F0 sensitive neurons. I change the F0, they change their firing rates or their firing spiking patterns. And we find these all throughout the auditory cortex, not in one particular area. And they've been well described in the auditory cortex of many species, ferrets, macaques, gerbils, and even in sort of human depth electrode recordings. But these neurons are also sensitive to the timbre of the sound, right? So they're not necessarily going to give you a readout that you can use to recognize, oh, that was the same melody on a violin and a piano. And so if you're looking for these types of pitch selective neurons, these have been described to date really in my mind, only really convincingly in the marmoset. So in the marmoset auditory cortex, Bender and Wang have described pitch neurons that are tuned to fundamental frequency of lots of different types of sounds, click trains, resolved harmonics, unresolved harmonics. And they find those in a special region of auditory cortex, in low frequency auditory cortex, and they call this the pitch area. But we've been unable to date to replicate this in any other primate or non-primate species. So I'm going to talk you through sort of 3 lines of research, and all of the research I'm talking to you about today are actually a part of the PhD thesis of an absolutely fantastically talented PhD student in our lab, Veronica Tarka. So in this first study, we want to ask, do ferrets have pitch-selective neurons in their auditory cortex? We know ferrets can do lots of different pitch discrimination and pitch detection tasks. So can we find any pitch-selective neurons in auditory cortex? And can we find any evidence of this dual processing of resolved and unresolving cues in the ferret? So to examine this, we did neural pixel recordings. We recorded lots of neurons in both regions of the primary auditory cortex in the animal. I'm going to be referring to neurons, and to keep Ellie happy, I will specify that by neurons here, I mean a well-isolated single unit, but who knows if it's a neuron? It's best we can do it with EPIS. So let's focus first on these 3 stimuli that I'm presenting. First of all, broadband click trains, okay? So they will contain both resolved and unresolved harmonic cues, and will vary their pitch. And we'll also present stimuli that carry the same pitch, but they have only resolved or unresolved harmonics as far as we can best model the ferret ear, and based also on some of our ferret behavior. So, oh yeah, that's the 3 sounds. So they sound different, they have different timbre, but you would equate them to the same musical note. All right, so let's look at these H neurons, these harmonicity neurons as we're calling them. So they need resolved harmonics to extract pitch, so they're going to be showing some sort of tuning to a fundamental frequency that's consistent across these first 2 stimuli, but are probably going to be unresponsive or certainly not pitch tuned to the third. We might then look for temporal neurons, right, which are going to be F0 tuned for the full broadband click trains, and the unresolved harmonics, unclear what they might do for the resolved harmonics. There's still pitch cues in there, but they're not as explicit in the auditory nerve. So we would definitely see tuning for those 2 stimuli in a temporal neuron. And then we might hope to find actually some pitch neurons that are able to be tuned to all 3 of these stimuli. So it's invariant to whether it's resolved or unresolved. All right, so what do we find? Well, we found all of these neurons in the primary auditory cortex of the ferret. Okay, so I'm just showing you one example of each here. So we have my nice sort of idealized models of these 3 neuron types up top, just to remind you, and below an example of each of these neurons, okay? So sometimes the pitch tuning is a bit complicated, like here it's almost tuned to 2 F0s, but you see that's really consistent even when I change the spectrum of the sound really a lot. And in the case of the pitch neurons on the right, these are even, sounds with non-overlapping spectra, and it's still showing the same tuning. So that's exactly what we'd like to see to be able to say these are pitch selective. So just like in the marmoset, these neurons are rare. You've got to go hunting for them and be a bit belligerent to find them. So we found about 165 neurons that were F0 sensitive to the click train, so they showed some hope of being F0 sensitive, and basically about 20% of those neurons were either harmonic neurons, but 20% harmonic, 20% temporal, and 20% pitch encoders. So if this is truly a harmonicity neuron, it's going to extract the pitch of not just these first 2 stimuli that I presented, but we can present other sounds to make sure these neurons can really generalize their F0 tuning across different stimuli, and that's what I'm showing you here. So here we're presenting now missing fundamental sounds and some click rates with a little bit of jitter in them, so that changes the spectrum and the tempo dynamics a bit, but not enough to, totally destroy our perception of pitch. And we see these consistent vertical bands means that the tuning is pretty consistent across these, again, very spectrally different, temporally different stimuli. And the bottom is just an example of a bootstrap analysis that we're doing to make sure the structure in this tuning is statistically significant. Okay, so, but are these really pitch neurons? So many people don't know that the 1990 hit horror film, The Skeptic, was actually a true story of an auditory psychophysicist. But there you have it. So we need to run some controls. Let's really make sure these are pitch neurons. She has smiling. So, are these neurons, first of all, sensitive to temporal regularity? This is a stimulus that Bender and Wang feel very strongly about. So if I present a click train, as I've been talking about before here, the clicks are perfectly periodic, right? And that gives you a nice clear pitch. But what we can do is just kind of jitter a little bit in time, either a little bit, 10%, or a lot, 40% jitter, the exact timing of each of those individual clicks. And what you get then is a sort of gradual breakdown in the saliency of the pitch that you hear. Right? So it's sort of going to a clear tone to noise in the 40% case. Well, that's to us, right? But we're not ferrets, are we? No? Stephen David was close. Okay. So if we go back to our ferrets that are trained to do a 2 alternative force choice pitch discrimination task, we see that they are also perceptually sensitive to this temporal regularity. Okay, so if I introduce more than, say, 20% jitter, their behavior goes to chance. So what about the neurons? So if we look now at the neurons that are F0 sensitive, okay, not necessarily selective, we find some neurons that are very giving away the plot line, very jitter sensitive, like this one up here, okay? So that is a really nice neural correlate of what we experience behaviorally, and it seems the ferrets experience behaviorally. So the pitch tuning is really consistent until I go above about 20% jitter. But actually we find a lot of F0 sensitive neurons that are more like this tuning curve on the right. So they have some sort of F0 tuning, or so we think, but as we disrupt the temporal regularity, they keep that tuning. So bizarrely to me, although the percept is changing immensely, right? You heard it. They're actually just keeping track of events per second. So they're basically giving you a readout of the click rate. So that's not very nice for pitch perception, right? Because we know our pitch is falling apart. So what do we see in our special neurons? Well, across the population of harmonicity neurons and temporal neurons, as well as pitch neurons that I'm not showing you here, we see this gradual decrease in the firing rate as I jitter the stimuli more and more. So it's, again, a really nice correlate in these types of neurons with the pitch that we experience and ferrets experience. A second control we might ask is, in the case of the temporal neurons, are they phase sensitive? Well, they should be, right? So if I, so far I'm presenting my harmonics perfectly in phase. If I disrupt that phase relationship, so over here in the randomized phase stimulus, each harmonic is now maximally out of phase, actually. It's not really random, it's sort of Schroeder phase. And what you get is like a flattening of the temporal envelope, right? Because the phases of those harmonics no longer line up. And if you're using temporal cues for pitch, that should destroy the pitch percept, which is going to be the case here because I'm presenting only high frequency resolved harmonics, so I'm forcing you to do temporal pitch. If we alternate the phase, we can present each harmonic in cosine, sine phase, and you actually get like a doubling of the periodicity of the temporal envelope. So you might expect the pitch to be twice as high as it kind of really is. And what's cool here is you see the spectra of these sounds are actually identical. So again, do ferrets care? Yeah, they care. We care, ferrets care about these phase disruptions. If I play the sounds for you, that you'd probably say they sound the same actually, but if I get you to do a pitch discrimination task, your behavior is going to suffer. And so it does with the ferrets. So if I present randomized phase stimuli, the ferrets and the humans, their behavior on this pitch discrimination task fall to chance. And here's an example from one of our populations of pitch neurons, which is a nice example because it's showing exactly what I hoped I would see. And that's a neuron that's tuned to pitch in the black curve, right? So it has some fundamental frequency tuning. If I alternate the phases, the neuron thinks the sound is twice as high, and so it's tuned to something twice as low. And if I present in green the randomized phase, it kind of doesn't fire very much because we now have a weak temporal pitch. And this holds up across our population of temporal pitch neurons. So I'm plotting here the kind of center of mass or the preferred fundamental frequency, and you can see it's lower for alternating phase than it is for sounds in phase, and the neurons respond less and less as I introduce these phase disruptions. Okay, and one last control is to make sure that these neurons are not actually responding simply to the energy in the cochlea at F0. So we know that when you present a harmonic sound, the cochlea can kind of start to wobble due to the mechanisms of the outer hair cells at F0. So to control for that, we use a standard control, which is to present the same sounds in the presence of a pink noise masker. So the mask is going to mask that small cochlear artifact that you sometimes get at F0. And what you see here for the harmonicity neurons and the temporal neurons is they maintain their F0 tuning really well in the presence of this pink noise masker. Also, a sort of entirely different way of controlling for this is to, instead of trying to hide the cochlear distortion product, actually just present a pure tone. And what happens is, unlike the neurons described in the marmoset, which were kind of classified as well based on their pure tone tuning, if we just go hunting for these cells tuned to complex pitch, they usually don't respond to a pure tone, okay? Because this is a weak input to a circuit that's set up to integrate across frequencies, right? You do experience a pitch, but it's just one frequency. So it's a weak input to these neurons as I envision them. And you sometimes get neurons that are actually tuned well, but to a very different frequency of tone than their F0 tuning, which is consistent for complex sounds. So in our mind, frequency tuning and pitch extraction are two very different things. And do I find them in a special pitch area? Wouldn't that have been nice? Finally send something to nature. Sadly not. Again, they're distributed. They're distributed all throughout the primary auditory cortex, these temporal and harmonicity and pitch neurons. So that's in contrast to the marmoset data that I mentioned, but it actually aligns really well with the data from other studies in other species, which include our work in ferrets as well as work by others in macaques, and also the representations of pitch that we see in human patients where we're able to put in depth electrodes, or other people are putting in depth electrodes. So yeah, there you have it. Maybe the marmoset is special. That's the best I can tell you. The data are the data. Okay, so we do find evidence for dual pitch processing and certainly pitch selective neurons in the ferret, almost just like we see in the marmoset. So this is good evidence that those are not pitch encoding in this way. Pitch specificity is not a primate thing. It's certainly something that other low frequency hearing mammals share. Whether you see it in a mouse, I don't know. Okay, so I'm going to talk to you now just more briefly about two other works of Veronica's. The first is looking at population pitch codes. So I went hunting, we went hunting for these special pitch neurons, but actually we want to ask now, if we just read out the population of neurons that are responding to these sounds, can we also get a robust representation of pitch that can generalize across these different timbres? And if we do that in this now more agnostic way, do we still see any evidence of dual processing of pitch, so harmonicity and temporal processing? So now we take our full sort of repertoire of stimuli, and we concatenate the neurons in our data set. And Veronica trains a pretty straightforward linear decoder to decode the pitch of the sound that she plays from trial to trial. But she's fitting this decoder separately for each of these different stimuli. So for each timbre, she extracts the pitch. And then we ask if we fit a decoder to extract pitch for stimulus A, can we read out the pitch of a different type of stimulus, stimulus B? So how sort of general are these pitch codes? Right, so we train and then we test on a different stimulus. And this is what you find. So what we're showing you here is sort of the training stimulus and up here across the sort of Y side of this matrix, and then how well it does on all these other stimuli. And this is not a random matrix. There's certainly structure in there that pops out at me. Hopefully it's popping it at you, otherwise I'll help you along. So some things of interest to note is that if you train on a broadband sound that's going to have resolved and unresolved harmonics available in it, it classifies pretty well across the board, with the exception of these random phase stimuli, which makes sense because actually you're not meant to be able to extract the pitch of those so well, and not doing so well on the pure tone either. We also see a group here of the unresolved harmonics, so there seems to be a population code there where you can generally use this to extract the F0 of unresolved harmonics. The tone stands alone, right? So the tone decodes the tone. Frequency decodes frequency. It doesn't decode pitch. Again, fitting with my sort of worldview of what's happening with pitch representations in the brain. And the unresolved harmonics, so this is our only stimuli with only unresolved harmonics, and this also kind of didn't generalize very well to the high harmonic stimuli here, suggesting that these are probably distinct mechanisms, okay? So with this more sort of, let's just see what we find approach. Another way to look at that is Veronica has taken the decoding weights and put them into PCA space and then clustered them, and you see again these nice separations of population codes that you can use to read out the pitch of either, you know, unresolved harmonics or broad spectral cues or pure tone, but not all of them together. And she has a poster on this population business this afternoon, or yeah, so if you're interested, please go to her poster and she'll be able to answer your questions about that work and give you more details. So finally, just to give you a hint for what we're going to be working on in the future, we want to now move towards using pitch as a selective attention cue. Okay, so not just looking at how neurons represent the pitch of a stationary sound. So as I'm sure you're aware, pitch is a very important cue for stream segregation or attending to a voice in a crowded room. We know very little about the single neuron basis for this process. How is it we're doing this? So that's what we want to be able to study in ferrets. So I might just, I'll give you my demo. Do we want a demo? Yeah. So I'll play you two sentences at once. And one sentence starts a little earlier with the best, okay? So listen to that sentence and follow it through and tell me what that person says, okay? The best.
The best ice cream has chocolate sprinkled right.
That was quite loud, sorry. So, who knows the whole sentence? Excellent. It's like super hard, right? So what I've done there is I've made it really hard because they're both my voice, and I've matched the pitch of those two sentences, so you have no pitch cue. So what happens now if I give you a little pitch separation between the voices? You ready? Okay, follow the best.
The best ice cream has chocolate sprinkled right.
Anyone get a little more of the sentence? Excellent, you young, healthy listener, you. Excellent, well done. Right, so it becomes a lot easier when you can segment the voices based on pitch. That's what I'm saying there. So we want to do a version of this task for ferrets, and one that's been used a lot in human psychophysics is, you present an A stream and a B stream, and the person has to detect like an oddball in the B stream, but not in the A stream. So you get an objective readout at how well they're attending to one auditory object, not the other. So in this case, we're training ferrets on this. They have to release a nose poke when I present sinusoidally amplitude modulated tones in the B stream, but ignore those modulations in the A stream. And if these are pure tones, it's kind of easy because they're on different parts of the tonotopic map. But if they're complex tones, you get overlapping representations. So can ferrets do this? And long story short, yes, ferrets can do this. But it's hard. Okay, so I just showed you a video to convince you that this is hard, but ferrets can do it. And as you can see in the data here, they do better when I give them more of a pitch separation for these complex tones. And I'm roving the A and we're roving the B, and they really just have to separate the streams. So that's what we want to work in next. Now we want to take these pitch neurons and pitch populations and look at how they help us segregate sounds. So that's coming to a grant proposal near you. Please be kind. All right. So the take-home message of the work basically is that there are physiological and behavioral characteristics of pitch that are shared among mammals, primates, non-primates, humans, ferrets. And I'll just end by giving credit to everybody that's worked on this in the red boxes here. And maybe there's time for one question. Thanks.
So, questions?
For a great talk. So I'm very interested in using pitch for speech-on-speech segregation, like in the example that you had at the end. In speech, pitch is typically on the lower side, maybe like 80 hertz to like 200, 250 hertz. Mm-hmm. And we know, we had a beautiful talk from Emily a couple of days ago, that in auditory cortex, we also have population responses at these frequencies. So my question is that something that you see in the ferret, that you've got time logging? So not, so to speak, pitch response or specific neurons per se, but neurons, or maybe population responses when you do pitch decoding that show kind of phase logging or time logging at the lower pitches, like maybe 100, 150 hertz.
Yeah, that's a good question. You would think there should be so ferrets are low frequency hearers, but in our behavioral tasks, once you go down to about pitches of 100 hertz, their behavior really starts to fall apart. So we tend to present F naughts are a little bit higher than that. So yeah, we don't see a lot of sort of phase locking to F naught, and most of what I'm studying and talking about are like spike rate codes for pitch. But they do have important and interesting temporal spiking patterns, but they're not phase locked. They're not synchronized. Yeah.
Okay, thank you.
Yeah.
Honestly, this is not a skeptical comment. I just noticed for the first time that confusion matrix that you had, which had the resolved totally segregated.
Yeah.
I'm trying to interpret it, and you can correct me if I'm, from the temporal. It seems to suggest that in the population, if you have a complex stimulus like a typical or something like that, then the temporal portions of the stimulus would give you basically a different pitch altogether from the resolved ones. There's no reason why they have to have the same pitch. They're parallel and independent. And obviously, this is nothing at all what we perceive.
Yeah.
So how do you interpret all this?
Yeah, so I'd say that's not really the way I'd interpret those graphs, because what we're doing, right, is we're giving the model just resolved harmonics, and then we're forcing it to do everything else. So if I give it those broadband click trains, it's pretty good with doing broadband stimuli and resolved harmonics and temporal, unresolved harmonic stimuli. So it can generalize if you start by training it with stimuli that have both those types of cues. It's just when we force it into picking out the resolved harmonics that they don't, the classifier then can't generalize to other stimuli. Does that make sense? Yeah.
Questions there? Yeah.
Really beautiful. I am thinking much more about pitch as it occurs in human speech. And there's two things about it that I've been thinking during your talk. One is that when we talk, there's slight continuous fluctuations in pitch. And I was wondering whether the kind of robustness to jitter that you've shown in your talk might underlie our ability to stream that. And the other is that these slight fluctuations, what they give us is prosody, and they're much more important is the dynamics, the relative pitch. So I was wondering whether ferrets have relative pitch. Or
Yes, okay. So first one is, does the jitter kind of help us with speech? Those who know me know I usually have a little bit of a clearer voice on this. Thank you, dance party. So yeah, I think having that little bit of robustness to a little bit of jitter is very helpful because real auditory objects are not perfectly harmonic, right? They have a little bit of jitter in them. So yeah, I totally agree. The second one, so when ferrets vocalize, they duke. It kind of sounds like right? So they have some prosody. Now, whether they interpret that in a meaningful way, we don't know. But lots of animals determine, how angry another animal is, how big it is, whether it's male or female, whether you want to mate with it, based on the pitches of the voices. So I think there are many sort of similarities or equivalents to prosody in speech in the animal kingdoms.
Amit?
Thanks for a wonderful talk, Kerry. I had a couple of different questions. First, with your decoder, have you tried separating into the different cell types, the different neural types, and seeing how that shifts, the decoding? And then the second question is more about organization. Like, I saw you were in sort of A1 and AF. Have you moved to AG or PEG at all yet? Or how much was there of those?
Okay, so I got the first one gone now.
Decoder.
Decoder, yes. There was the decoder, the question was, what happens to the special neurons if we decode them? So it's going to do well, right? You give better input, the decoder's going to do better. So that's why I think with decoders, it's really more the comparisons across conditions that are interesting, right? Because we could also make this a nonlinear decoder and more clever, and it'll do probably better, right? But what we have done is kind of the opposite, which is interesting. So if we exclude our pitch selective neurons from the decoder, can it still do anything? And it can with some stimuli, but not others. So it takes a little bit of a hit, right, because you're taking the best neurons away. But the pure tone decoding does not change when we take out our special pitch neurons, which again makes me feel pitch is not frequency in the brain or in our percept.
Okay, last question.
And we haven't gone into those higher orders areas yet with this, but definitely want to.
Was there a comment about any role of potentially spectral combination sensitivity in the ferret auditory cortex? And if it might have a role here?
Yeah, so that's what if I understand your question right, that's what we think the harmonicity neurons are doing, is they're taking the spectral content and like integrating it and looking for these special relations where they're all multiples of some fundamental frequency.
So basically, each neuron would have, like, say, more than one excitatory response area, and then super additive effect.
Yeah, and maybe inhibition in between the harmonics. It's all very theoretical at the moment, but that's how we're thinking about it.